Welcome to the BioInsights, where science comes alive.
Today, we are going to completely demystify spectrophotometric data analysis. Seriously, if you've ever stared at a spreadsheet just packed with raw lab data and thought, how on earth do I turn this into actual meaningful numbers? Well, you're in exactly the right place. We're going to take this whole process and make it clear, simple, and honestly, highly efficient.
So here is our roadmap for today. We'll start with the unknown sample mystery, move on to plotting your standard curve, then unlocking the trendline equation. After that, we'll cover method 1, which is the equation, followed by method 2, the trend function, and finally, we'll wrap up with some pro tips for accurate results.
Okay, part 1, the unknown sample mystery. What exactly are we trying to solve here?
I want you to think of this as a sort of detective story. Your lab equipment just handed you a major clue, a raw optical density reading straight from the spectrophotometer. Maybe it's 0.428 or maybe 0.76. But, what does that actually mean? Because without context, those numbers absolutely do not tell you the actual concentration of protein or phenol in your test tube. So the big question is, how do we find an unknown concentration from a raw absorbance reading?
Think of your data like a clue board. You have your knowns, your standards. Let's say you deliberately prepared these with known concentrations of 20, 40, and 60 milligrams per milliliter, and you got their absorbance readings. That's great. But then you've got your suspect, the unknown sample. You know its absorbance is 0.760, but its concentration? Total mystery. To put this all into perspective, let's walk through the actual chronological lab-to-data timeline. At minute 1, you pull your raw spectrophotometer reading. By minute 5, you're sitting at your computer, averaging your replicate readings, your R1, R2, R3 in Excel. By minute 10, you're visualizing the scatter plot. And by minute 15, you're calculating the final concentration. It's really helpful to know exactly where our Excel magic fits into your physical lab workflow.
Part 2. Plotting your standard curve. Time to build that clue board.
So the crucial thing here is moving quickly and cleanly through Excel. First up, you just select your data range with your knowns. Second, hit the Insert tab and click the Scatter Chart option. Third, right-click on literally any of the dots that pop up, select Add Trendline, and make sure you pick the Linear option. And fourth, actually scratch that. This is the most important step. Make sure you check the boxes for Display Equation and Display R-squared value. Boom, just like that, your standard curve is built.
Part 3. Unlocking the trendline equation. Our magnifying glass.
You might be wondering, why do we even need a trendline in the first place? Well, it acts as our mathematical magnifying glass to predict values we haven't directly tested. And a massive part of that is the R-squared value. This is a statistical measure representing how close your physical data points are to that fitted regression line we just drew. It's essentially the ultimate quality check for your lab work. Ideally, you want a value that's very, very close to 0.99.
And to really drive home why that matters, the source material explains it perfectly. If the R-squared value is around 0.99, it means the data points aren't too scattered. The trend line is highly reliable. In fact, one of the examples from our sources yielded an R-squared of 0.9909. When you see a number like that pop up in Excel, you can take a breath because you know your assay data is fantastic and your pipetting technique was pretty much flawless.
Part 4. Method 1. The equation. Basically, algebra made easy.
Now we actually have two different paths to solve our mystery, starting with the manual equation. When you checked that display equation box earlier, Excel gave you a raw output looking something like this: y equals mx plus c. Let's visually break this down so it's crystal clear. Y represents the absorbance reading you already have in your hand. X represents the concentration. That's the unknown piece of the puzzle we're hunting for. M is the slope of the line, and C is where the line intercepts the axis.
Since we need to isolate our unknown concentration, which is X, we just do a quick magic trick. We rearrange the formula. By shifting the variables around, we get the algebraic flip. X equals Y minus C, all divided by M. See? It's just straightforward math. No complex magic here.
Let's anchor this in a real-world example from the source data. Let's say your unknown sample gave an absorbance reading of 0.76. We take our arranged equation and just plug in the values Excel gave us from the trend line. So our y is 0.76, we subtract our c value, which is 0.0133, and then we divide that whole thing by our m, slope, of 0.0115. You type that little formula into an Excel cell, hit enter, and bam, 64.93. There it is. We have our final deduced concentration of 64.93 milligrams per milliliter. We completely solved our initial mystery, translating a totally confusing absorbance reading into actionable, real scientific data.
Part 5. Method 2. Trend function, aka the Excel shortcut.
Now, that first method is great, but what if you're working with a massive 96 well plate and have literally dozens of unknown samples? Doing that algebra over and over? No way, that's going to take forever. Let's juxtapose our two approaches so you can pick the right one. Method 1, the equation, requires a little algebraic rearrangement, but it's absolutely excellent for visually checking things on a graph. Versus Method 2, the trend function. This is a fully automated Excel formula. It's incredibly fast for bulk samples, and get this, it technically requires absolutely no graph at all, so you can choose whichever method fits your specific workflow.
Let's look at the syntax for the shortcut. You type equals trend and open a parenthesis. Excel is going to ask for three things. First, you highlight your known underscore y's. Those are your standard absorbances. Next, you highlight your known underscore x's, your standard concentrations. And finally, you select the new underscore x. Now pay super close attention here. In this specific context, the new underscore x is actually where we input our unknown absorbance reading. Once you select that cell and hit enter, the formula literally does all the heavy lifting for you instantly. It's a real game changer.
Part 6. Pro tips for accurate results, ensuring lab perfection.
Okay, listen, a brilliant Excel formula can't fix bad lab procedures, right? So here's a quick summary of actionable tips to make sure you don't just get an answer, but you get the right, scientifically valid answer. First, always average your replicate readings before you do anything else. Second, if you diluted your sample to get it to read properly, do not forget to multiply your final answer by the dilution factor, like multiply by 2 for a half dilution. Third, ensure your data arrays are equal in length and linear. Otherwise, Excel is going to throw a hashtag value or hashtag ref error at you. And finally, beware of excessively high absorbance readings. Anything above a 2.0 on a spectrophotometer is generally just not reliable. Well, you've got the full toolkit now, so I want to leave you with a question to think about. How are you going to apply this standardized Excel shortcut to speed up your next lab experiment? Whether you lean towards the visual equation method or you go all in on the automated trend function, you are now completely equipped to conquer your data. Thanks so much for joining me on this explainer, and hey, happy analyzing.